We'll go over a few slides just to remind you of some of these topics that are important, okay, and to help you guide your studying. All right, so we were in the last third of the class semester. We began to shift our focus away from just understanding all of the intracellular pathways like oncogenic pathways, tumor suppressor pathways, that become dysregulated inside of a cell as part of transformation. Right? And a key part of that was understanding how all of it kind of has to do with mutations that happen in the genome that result in alterations in those types of critical pathways that ultimately result in the cell losing control over proliferation. So then we shifted then to a discussion about the relationship of that transforming cell with the surrounding tissue. And we began to appreciate how a cancer can only get so far in terms of developing into a large, out of control proliferating tumor mass before issues of the surrounding tissue become highly relevant in determining whether that tumor can continue to grow and ultimately spread. So we began our discussion by understanding that the extracellular matrix, or the material surrounding a cell as part of the tissue that the cell is normally associated with, is this big mix of different types of proteins and different types of signaling molecules. And this provides not only a sort of reservoir for different types of signaling molecules, but also structural support. And with our discussion of epithelial cells, which have been the main focus of modeling cancer this semester, is just looking at epithelial cell cancers, right? The basement membrane is kind of like a specialized extracellular matrix region that's immediately underneath layers of epithelial cells. So this cartoon, I think, is a nice one because it allows you to appreciate that many of these protein components of the basement membrane, right, are these long types of collagen fibers or laminin fibers that create really sort of like a molecular, cloth, if you want to call it that, like a fabric, right? That you can really understand can provide anchorage, physical anchorage for epithelial cells. So you'll recall that in this cartoon here, the gray double layer here is the lipid bilayer membrane of the cell. So anything below this in the cartoon would be inside of the cell, and anything above this would be outside of the cell. And we said for an epithelial cell, it would be attached to this basement membrane, and it associates with that membrane through different types of cell membrane receptors, like these green things poking out here, which are meant to be the integrins. And so we learned that the integrins, because of their shape, they can properly interact with certain components of the basement membrane. So such as in this corner down here, right, these blue laminin molecules are being attached to these integrins that are sticking up. And as a result of that protein-protein interaction, right, the shape of the green integrins is kind of altered a little bit. And so on the inside of the cell, the other end of this molecule that sticks into the cytoplasm, that shape will also be altered, right, because of the allosteric changes in the protein shape. And that then will signal the epithelial cell through a series of different proteins that can interact with those altered shapes, right? And ultimately tell the epithelial cell you are properly, happily attached to a basement membrane, so you should go about business as usual, right? And maybe carry out your normal epithelial cell functions. So anchorage is a big part of the basement membrane, but along with anchorage, that also means confinement. Right? The cell is kind of pushed up against this physical boundary of the basement membrane. And just like imagining this being like a cloth or a fabric, right? Lots of mitogenic signaling molecules will get trapped in here and not quite make it to the surface of the epithelial cell, right? So storage of mitogenic signaling molecules and limiting the amount of those molecules that actually arrive at the surface of the epithelial cell is another important aspect of the basement membrane. And I think by now, I think all of you can appreciate, right, when we talk about ultimately the cancer cell or the tumor becoming metastatic and breaking through this basement membrane, why those types of issues then become very important. Okay. So part of the tumor growing and needing to be able to manipulate the stroma around it, this is the idea of the rebelling teenager who gets more and more, angst-ridden or whatever because, he or she hates her life because her parents make her miserable. How she manipulates her parents, and it creates a dysfunctional household that ultimately, if things keep progressing that way, result in not only the cancer cell wanting to escape from the confinement of the original in situ tumor that it grew from, but also its ability to escape with the kind of manipulated help of the stroma around it, what we call the reactive stroma, right? That's reacting to different types of messages that the tumor cells are sending out. So remember, one of the key factors or key series of events of a tumor being able to grow and actually go beyond the boundaries of its basement membrane confinement, right, is tricking the surrounding tissue to activate the sort of pre-programmed wound healing response. And so this flow chart here, I think when we first saw it, I explained to you that this is primarily the flow chart that you would look at if you're just talking about straight ahead everyday wound healing, right? You get a cut on your arm and these things happen. And importantly, these things are pre-programmed to happen rapidly and with not a lot of fancy delayed upregulation of different types of proteins and things. Because evolutionarily, if you cut your arm and you're bleeding, right, you need to have a whole series of things happen quickly. Otherwise, you can't sit around for 6 or 8 hours as you're bleeding out from your arm. So evolutionarily, we developed these wound healing responses. And as it turns out, if the tumor cells, because of random mutations that they stumble upon, learn how to activate this wound healing response, there's a great irony there. Because this wound healing response was evolutionarily pre-programmed to help us, right, by healing our wounds. But in the case of cancer, it's actually used against us because it's used by the cancer cell tricking the surrounding tissues to actually do things that ultimately help support the cancer. And so for those of you who wonder, like, well, that's really weird, because how could you have such an important series of pre-programmed events like this wound healing be part of our evolutionary fine-tuning, and yet somehow this is like a big thing that can be tricked by cancer? Why does Mother Nature doesn't account for that? Again, remember, it's because Mother Nature never really figured that we would have to deal with cancer as a problem, right? That for the 40, 45 years of a normal, what Mother Nature expects our lifespan to be, right, it's more important to have a good wound healing response for your everyday sort of little accidents and mishaps, right? It's not so much a big deal worrying about, oh, but cancer can actually use this to its advantage. So look through this, remind yourself of the parallels. What's happening to the stromal tissue? For example, damage to vasculature as a result of, let's say, a cut or an injury, normally resulting in things like platelets being attracted, platelets being degranulated. That means platelets releasing certain types of stimulatory growth factors like platelet-derived growth factor, or PDGF, right, or TGF-beta. the conversion of certain types of extracellular matrix proteins, for example, fibrinogen to fibrin, right, to form clots and things like that. These are normal parts of wound healing at the beginning, right? And how the growth factors that are released, like for example, the PDGF, result in things like the recruitment of macrophages that normally would come and start remodeling the stroma to get rid of the damaged tissue, get that out of the way to allow for new healthy tissue to grow back. And that would then result in things like macrophages using matrix metalloproteinases or MMPs to digest away the extracellular matrix. That may also involve digesting away the basement membrane, resulting in the release of many of those growth factors that were sort of stuck in the cloth or the fabric, right? So now you have a big increase of mitogenic growth signals, which again, in the context of wound healing makes sense, right? You suddenly want to encourage epithelial cells to begin to divide and grow and even migrate. Part of this also is the upregulation of things like VEGFs, right? The vascular endothelial growth factors that begin to stimulate nearby capillaries to begin to branch off and start growing in the direction of the wound, right? Because you have the damaged, compromised circulatory system there at the site where you have all these capillaries that might have been cut or shredded, right? Now you have new capillaries that almost right away begin to start growing in that direction, right? And that process of stimulating new blood supply growth is, of course, what we call angiogenesis. Right? So this results in new capillaries heading in that direction and epithelial cells proliferating. So if you go to this part of the cartoon here, if you just think about these epithelial cells, they have to undergo epithelial mesenchymal transition, or EMT. So the nearby epithelial cells that maybe weren't damaged in the cut, right, they have to be encouraged to let go of being normal epithelial cells, turn into more mesenchymal-like cells so that they can begin to migrate through the stroma and begin to repopulate the site to build up new tissue. So they move into the wound site, proliferate, they cover the wound, and importantly, what? After they do so, they have to revert back to being their normal epithelial state. So the reverse of EMT is mesenchymal epithelial transition, right, or MET, and that's what reconstructs the epithelium. So I think the amazing thing about this is this flow chart, right, is what applies to everyday wound healing. But now you realize that these series of events, right, if a developing, growing tumor can trigger these events to happen, right, these types of remodeling and encouraging epithelial cell transition to being more stromal-like, right, they all end up favoring metastases of a tumor. We looked at examples of how different types of stimulatory factors are important for providing the tumor certain ways out of limitations or challenges that a large tumor is facing. One of the most important is, of course, the angiogenesis, the encouragement of capillaries to provide more blood, more oxygen, more nutrients to the ever more demanding population of tumor cells. We pointed out that oftentimes solid tumors, when they get too big, then the cells at the center of that tumor are cut off from oxygen and nutrients, and they die. And that's why the insides of large solid tumors are oftentimes, you know, I said it before, they're kind of nasty, right? Because they're all necrotic, dead tissue. And there's a lot of times a lot of fluid, stuff like that. So if the tumor cells can turn on VEGF expression, then maybe they can go ahead and recruit things like new capillaries to grow. And this was just a reminder of how things like the VEGF gene, the vascular endothelial growth factor gene, are regulated by proteins that can detect things like normal oxygen levels. Right? So remember, one of the important Transcription factors are these HIFs, like the HIF1A, HIF1B. Normally, right, in normal oxygen levels, there is another enzyme, this proline hydroxylase, that if there's plenty of oxygen present in the cell, so in other words, normoxia conditions, this hydroxylase will use that oxygen and modify or put these hydroxyl groups on the HIF1A. That altered shape of the HIF1A is recognized by the tumor suppressor gene, the VHL, which then, that's the red thing here, which then binds to it. It acts as a ubiquitin ligase that then begins to put lots of ubiquitin molecules on the HIF protein, which leads to it being targeted for degradation, right, and recycled. So its amino acids and peptides can be used again. But under hypoxic conditions, meaning low oxygen conditions, you'll have more and more of these HIF molecules escaping this polyubiquitination and degradation. And that means more and more of them will successfully make it to sites in promoters or enhancers of target genes, such as VEGF, right? And that will then upregulate the expression of VEGF, and that VEGF then might then begin to stimulate capillary growth from outside of the cell, right, in the surrounding stroma, which would then benefit the growing tumor. I love this illustration because I think this is a very clear illustration. Here you have this benign tumor here, so a benign mass, right, of tumor cells that are still confined by their basement membrane. So this arrow here, right, you kind of visualize a basement membrane here. And immediately on the outside of this basement membrane, you have capillaries. And so in this case, this is the maybe closest nearby capillary, or one of several, right? Any of the brown things you see over here. So for example, here, here, and here, and here. Remember how surprisingly extensive the capillary network is throughout our body's tissues. Right? We really don't appreciate that because, again, when you look at a typical human anatomy like chart, right, of the major arteries and things like that, you know, they never bother to go down to the level of the teeny tiny capillaries because that would just be, you know, too crazy of a chart. And not only that, but remember the major blood vessels in the body, like the aorta, or different types of smaller arteries that go to the extremities, right? Those are all laid out by genetics. That's why everybody's aorta is in the same place, right? The major arteries that go to your arms and legs, everybody has them. But the capillaries are improvised, right? So in this case, we have the capillaries that are going through here. Remember, a typical cell in the body will not be more than a couple hundred nanometers, or maybe it was 0.2 micrometers or so, away from the closest capillary, right? Because that's limited by how far the gases can diffuse and how far nutrients can diffuse back and forth from the circulation into surrounding tissue. So in this case, we have this tumor mass that's growing. It's getting a little bit too big now. That means the cells in the middle are starting to stress out. But some of these cells have learned to start expressing VEGF. And so as a result, maybe there's like a cloud of VEGF diffusing away from this tumor here. And some of it has reached the cells of this capillary and have stimulated now the capillary to grow in this direction, in this branch. But the important thing is what? This is still an in situ tumor because this tumor mass is still confined by its basement membrane. And you can see, just like the tumor cells themselves cannot break out of the tumor, likewise, the capillaries at this point cannot grow in either, right? So this tumor is kind of at this like stalemate right now, but it's kind of scary because maybe a few more mutations and some of these cells will then learn how to do things like switch on the wound healing response, to get things like macrophages to help out and digest away the basement membrane, right? And once that happens and the cells begin to invade the surrounding stroma, right, then they might really undergo that angiogenic switch and suddenly get tons and tons of capillaries growing throughout the tumor mass, allowing it suddenly to grow very large. So that means these tumors must trigger this angiogenic switch in order to keep growing. And I pointed out to you that limiting the vascularization of our tissues is another way that we evolved, right, to put brakes on tumor formation. Now, it might be a little misleading because you think, well, you know, the limitations on the growth of capillaries have a more practical kind of consideration, because if in reality we have this crazy extensive network of teeny tiny capillaries through all of our tissues, and that's improvised, that means it grows where they're needed, right? They cannot keep growing and growing and growing and branching out all the time. They need to know when to stop. Right? Otherwise, you know, things just get too crazy. So normally you think limitations on the growth of capillaries so that there's just enough to accomplish what our tissues need, right, isn't so much a way of preventing tumors to grow. It's a more practical thing of just keeping the crazy mess of our capillaries limited. Right? So here you have normal skin with the dotted line here represents maybe the basement membrane. The cells above might be the dermal cells stained in blue, right? And you have a few capillaries in cross section that happen to be caught in this microscope slide. So those are stained in red. As you get tumor development, so you get hyperplasia. So the transformation of the epithelial cells, the layer gets thicker and thicker, right? But they're still very clearly here, right, contained by the dotted line where the basement membrane is. Right? You know, maybe there's some additional capillary growth here. It's not clear. But certainly, once you get to the point where you get heavy dysplasia, then you really begin to sense that the basement membrane is being threatened here. Maybe in some places it's already broken, right, digested away, and the tumor cells might already be infiltrating the surrounding stroma. But really the thing that stands out here is the increase in the number of capillaries now that have been encouraged to grow in this area, providing the tumor what it needs in order to keep dividing. We studied the series of events that give rise to metastases as part of this so-called invasion metastases cascade. And we said we can think about it in kind of simplified terms of 7 steps, right? So the primary tumor formation that is happening here, so just like we saw in some of those images, maybe some of the cells are learning how to attract capillary growth to support the aggressiveness of this tumor. That leads to localized invasion. So now some of these cells maybe are breaking free of the basement membrane. They are now working their way through the surrounding stroma. So go back and remember some of the types of things that must happen for these epithelial cells to give up being epithelial cell-like and transitioning to being more mesenchymal. Things like the downregulation of E-cadherins on their surface. The E-cadherins that normally keep epithelial cells pushed up against each other, right, and signal each of those cells so that they know that they are happily with their epithelial cell neighbors, so that they shouldn't divide anymore. They should just mind their own business and do their job, right? How the loss of E-cadherins due to transforming mutations might be accompanied by things like the upregulation of N-cadherins. N-cadherins are what stromal cells use to be able to properly talk to each other, the stroma. And so if you have these tumor cells that now begin to upregulate N-cadherins, how that actually encourages them now to mingle, right, and hang out with stromal cells more than their original epithelial cell buddies, right? And so that encourages them to make their way to the wall of the closest capillary. One thing I wanted to remind you is this little zoomed in region here, even though it makes it seem as though, oh, this is the tumor mass and this is a cell making its way, you know, towards the closest capillary. Remember, if you go back to this first image here, at a certain point, there's going to be capillaries all throughout the tumor, Remember that one image I showed you of the mouse brain that in one corner had the tumor, and then they stained so you could see all the circulation, the vasculature, and how crazy messy the capillaries were in that one spot, right? So that means within the tumor itself, the tumor cells don't have to go far at all in order to actually gain access to a capillary, right? So that's why it's not just like, oh, one little tumor cell here, one by one, is going to somehow come into this capillary. No, in a large tumor, you may have at any moment hundreds of thousands, right, if not millions of tumor cells all entering capillaries at the same time. Right? So this process of entering the blood supply, we call that intravasation, and this is where they will interact with platelets and other things that are in the blood. Remember, they will clump together, and the platelets, even though they are, trying to form a clot because they think it's a wound, right? In reality, the platelets are protecting the cancer cells, almost like a shield, so that as the cancer cells are being moved through the circulation, something that probably the original normal healthy epithelial cells never were built to handle, right? The cancer cells can survive more. They get transported through the circulation, right? So maybe they make their way to, let's say, the heart, and then they get pumped out again. First, they get pumped into things like the lungs. Remember, once these clumps of cancer cells, which we call circulating tumor cells or CTCs, get lodged or wedged into different capillaries, right? Because the capillaries are really thin compared to maybe this clump of tumor cells that's jammed up against it, right? A lot of the tumor cells get sheared apart, right? But cancer cells are remarkably resilient in the sense that oftentimes a cancer cell can lose most of its cytoplasm and just have its humongous dysfunctional nucleus kind of still present, and it can survive, right? So then it might come free, or it might just stay stuck in the lungs, but let's say it comes free, and then it makes it somewhere else in the body where it then gets stuck, and then because it's now far away from its original highly dysfunctional reactive stroma environment, right, that it helped create over many years of interacting with the stroma, because it's far away from that now, it's kind of like chilling out, right? It's like the teenage runaway who's all hot and angry and angst-ridden and runs away from home to get away from his or her parents. But by the time a couple of days have passed, right, they're just really homesick and they want to actually return to a more safe, comfortable, familiar type of environment. So the tumor cells are like that also. So they get stuck randomly in the body, and they may extravasation, right? They leave the blood supply and they form a micrometastases. And remember, it may be that they ended up in some other stromal tissue in the body that is just way too different than the stroma that they came originally from, right? And as a result, because these cells still have the baggage or the memory of being, whatever normal type of epithelial cell they were, right? That means they may be responsive or expect different types of signals from the stroma in order for them to really resume being a happy epithelial cell, right? I mean, they're not really happy because, of course, all of their genomes are messed up here after all the mutations they've had. But in very rare cases, relatively speaking, right? Relatively speaking, meaning if you have hundreds of thousands of these micrometastases spread all around the body, right? Rare might mean you have a few hundred particular micrometastases that actually end up somewhere good for them, right? And those might then begin formation of a macrometastases. Right? So this has to do with the seed and soil model of metastases, which I think is a wonderful model because to me it makes a lot of sense, right? And that is to say that if you have a handful of seeds and you wander around the UH campus just randomly throwing seeds in different places, like next to a sidewalk, in the corner of a parking lot, or on a patch of dirt, the soil that has other plants growing already, and then you go back 2 months later, you're not surprised, right, that in some of those spots you get nothing. But in other spots, oh, now you have nice flowers, right? And so that's the seed and soil metastasis, idea of metastases. So many disseminated tumor cells, they survive as micrometastases in the new home, but they don't flourish because the soil is wrong. Right? And so therefore, in order for the emergence of a macro tumor metastases, it has to be a combination of not only the right seed, meaning the tumor cell itself has to have some ability to manipulate the new stroma, but the new stroma itself has to be that good soil that's appropriate for the disseminated tumor cell to be able to really take off and begin growing again. Right. And we said, importantly, different types of tissues in the body tend to be better soil for a wider range of metastatic lesions to form. Things like the bone marrow, for example, right? Because the bone marrow is highly conducive to cell proliferation because of its function, right? It's where all your hematopoietic cells come from, right? All the stem cells that give rise to your blood cells. So tons of mitogenic signals in the bone. So that's why oftentimes, right, metastatic lesions are in the bone. Right? Or even things like lymph nodes. Lymph nodes because, right, lymph nodes, it's more of like a convenience. Remember, we learned that from a tumor that's shedding tumor cells, cells might end up in the blood, but cells might also be drained into the lymphatic system surrounding our tissues or surrounding the tumor. And therefore, lymph nodes are oftentimes places where a clinician, when following up with a patient, might take biopsies not only of the primary tumor, but also they might sample some lymph nodes that are in the vicinity, right? Just to see if there's any evidence of cells being shed into these lymph nodes. So this cartoon, again, summarizes this concept of EMT and MET. So it's very important to understand, right? This process goes both ways. I don't think we need to go over right now. This is just put up here to remind you that you have this nice kind of overview cartoon. So talk your way through it. Remind yourself of the different events that happen along each step. Okay. We then shifted towards a discussion of our immune system and how our immune system represents our second line of defense against cancer, right? So our first line of defense, remember, is we depend upon the transforming cell itself to detect that it cannot make the necessary repairs to overcome whatever types of genomic damage that it got through, either a mistake in DNA replication, right? Because remember, the enzymes that replicate our DNA, they are not foolproof, right? So every time a cell divides in our body, you actually get additional mistakes left over in those daughter cells. That's one of the reasons why fully differentiated cells, we evolve so that they don't stick around, right? So that they don't have this baggage of mutations that then, if they were to keep dividing, they would just keep piling up these mutations. So our bodies are okay with the fact that a fully differentiated cell maybe only divides 3, 4, or 5 times more before it undergoes apoptosis or is just physically lost. Right? But if for some reason a damaged cell does not undergo apoptosis on its own, so normally the P53 would be putting the brakes on the cell cycle to allow time for DNA damage to be repaired. But if there's still too much DNA damage, right, P53 is continuously told to activate, right? It never gets the all clear. And eventually, as you have more and more P53 activity sustained, right, it becomes less an issue of trying to repair the genome. It becomes P53 hitting the nuclear button, right? And just saying, forget it, the cell needs to apoptose. Right? So the scary thing is that's what's happening in your body right now, right? At this very moment, you probably have hundreds of thousands of cells who are on the verge of transforming but they will do the right thing and undergo apoptosis, right? So that at the end of the day, those cells are gone. So what we worry about is, what if the mutations or the damage to the genome in these cells actually impacts those pathways that allow it to do the right thing, right? Because after all, those apoptotic pathways, which we call intrinsic apoptosis, meaning the message for apoptosis is coming from within the cell itself, if those pathways are all regulated by proteins, and the DNA damage has compromised the function of those proteins and those pathways, then you got a problem, right? Because maybe the cell won't undergo apoptosis. So then it's up to our immune system. As this cell begins to divide and form a tumor, our immune system needs to step in, recognize it as something bad, and tell those cells to undergo apoptosis. So instead of an intrinsic apoptosis message coming from within, a tumor, right, that's growing needs to hear extrinsic apoptotic messages coming from immune cells, like T cells or natural killer cells. So in order for the immune system to see that you have a tumor cell there, right, it needs to look for evidence on the outside of the tumor or the tumor cell. So oftentimes there are certain antigens, meaning certain three-dimensional proteins, things like that, that are either unique to just tumor cells. So they represent things like mutated versions of proteins that are now being shown off by the cell that the immune system, right, would recognize and be sensitive to. Or it might be that the proteins that they are displaying on the outside of the tumor cell are totally normal, healthy cell proteins, but the amount of those proteins is way out of whack, right? Or that particular protein should not normally be expressed in this type of cell. So that's why we have tumor-specific antigens that are unique to cancer cells, but a lot of the times we rely upon tumor-associated antigens, or TAAs, right, that just indicate that there's something not quite right with this particular cell. So these were two examples of TAAs. So in both of these slides, so we're looking at the skin here. So these are epithelial cells, keratinocytes here. On the left is your outer epidermal layer, the multi-layers of dead, keratinized epithelial cells. And mixed in with these epithelial cells at their basement membrane, you have the melanocytes, right, that produce melanin to protect the keratinocytes from UV damage. And so this particular red stain that's being used is staining for a tyrosinase antigen that these melanocytes use, right? So you can actually identify these melanocytes. So that means if this melanocyte were to actually become a tumor cell and wander away from here, then you would end up with a cell that's expressing a lot of this tyrosinase antigen somewhere else in the tissue that normally your immune system, would not expect to see a cell that has this tyrosinase on its surface. So that can be used as a sign that there's something wrong. Likewise, this red stain over here is staining for this MAGE-1 protein that's specifically made by the developing sperm cells in the testes. Right? So if you have a tumor forming here and eventually you have some of these cells entering the circulatory system and getting stuck in other places in the body, the fact that they have this spermatogonia specific MAGE-1 on their surface, if your immune system sees this in a cell that's in your lung, right, then your immune system might realize, oh, this is not right. And it might take care of that cell and tell it to undergo apoptosis. So this table was just a summary of different ways that our immune system is fooled by developing tumor cells as part of those tumor cells beginning to really make their way to becoming full-blown cancer. Right? Because remember, in order for a tumor to successfully become cancer, by definition, it means it has to have learned how to evade or get around the set of different strong mechanisms that our immune system has for properly detecting them. So I'm not going to read through this entire table, but let's just go through this left column here, right? And then it's up to you to go back and try to understand and appreciate things like the mechanism. So tumor cells can hide their identity from the immune system by doing things like no longer presenting proteins on their surface for immune cells to see. Those antigens might include different types of proteins that normally a cell that's undergoing a lot of stress, meaning damage or some sort of issues, critical issues having to do with not being correct, right? There are certain proteins that are actually expressed on the outside of the cell to indicate that the cell is stressed out and there's something wrong with it. So maybe these tumor cells can hide those as well. Tumor cells might be able to come up with mutations that inactivate different effector cells of the immune system from successfully being able to see the tumor cell. Tumor cells can avoid apoptosis. So again, they might be able to stumble upon mutations that disable those caspase pathways inside of the cell that are important for apoptosis to happen, right? So even though an immune cell has docked on the outside of the cancer cell and is sending cytotoxic messages saying, hey, you need to activate your apoptosis pathway, maybe some critical proteins in the apoptosis pathway are just missing because of mutations. And so therefore, the cell does not undergo apoptosis. The flip side of this is that maybe the tumor cells learn how to cause immune cells to undergo apoptosis. Tumor cells can also learn to neutralize different types of toxins. So we talked about things like upregulating different types of pumps to remove cytotoxic messages. They can neutralize complement proteins. Remember, complement proteins are those proteins in our immune system that can bind to the surface of a cell target and do things like make it easier for larger immune cells like macrophages to come and attack the target. Or even complement proteins can result in holes being poked in the surface of a tumor cell, resulting in the tumor cell just kind of imploding, right? Or I guess exploding because water would rush in. Right? We learned that certain proteins on the surfaces of cells actually inhibit the function of these complements, like CD59, for example. Another closely related one is the CD47, right, which also can discourage phagocytic cells like macrophages from eating the target cell. So go back, take a look at these, and just the take-home messages, right? All of these are different potential methods that will work in combination for a cancer tumor to be able to avoid the immune system. But the remarkable thing is, right, it's not as though the tumor cell itself, picks and chooses or has some sort of any kind of intention or clever ability to do these things. It's all, again, just by random mutations selecting for the subpopulations of tumor cells within a large tumor, right? You're going to have lots of different subpopulations forming because each of them are stumbling upon different mutations, right? So you'll select for those subpopulations that come up with these tricks. So as part of discussing the immune system, we discussed how antibodies are a big part of how the immune system can be leveraged to fight cancer. Right? So the last part of our semester, we moved into therapies for fighting cancer. And we said that the design of novel chemotherapeutics, drugs that target cancer cells, provide the most options, right, for new ways of targeting cancer cells compared to just, better surgical or better radiation techniques, right, that don't really discriminate necessarily between types of tumors, because that's not what surgery and radiation is primarily focused on. Those types of approaches are just about killing, right, tumor cells, right, with sort of either physically getting them out of there or just by high doses of radiation. Right? But chemotherapies, you can actually specifically target tumors. And that requires then that you have an understanding of the biology of the tumor such as what type of normal cell the tumor originally came from, because that will allow you then to come up with drugs that target specific pathways inside of the cell that might still be active and important in that particular subtype of cancer, or by targeting different types of antigens on the surfaces of tumor cells, so that the immune system that previously was ignoring the cancer now can be told or retrained to pay attention to it. So monoclonal antibodies are a great example of that. This figure was just giving you 3 examples of these important tumor-associated antigens, these EGFRs, right? These receptor tyrosine kinases that become maybe heavily upregulated in certain tumors, or the proteins, the receptors are mutated in such a way that they're always turned on. Right? So you can design different monoclonal antibodies, here's an example of 3 of them, that are actually designed to recognize different epitopes or different 3-dimensional features of the same receptor complex. And that gives you a lot of flexibility then to be able to target a specific cancer with maybe multiple antibodies. We also discussed how these antibodies are named. So remember now, every time you see a drug that ends with a MAB, you now know, right? It's a purified monoclonal antibody drug. But aside from antibodies, we can also develop new non-protein compounds, right, that can function because they can get into the cell and actually interact with different proteins inside the cell to intentionally interfere with different types of gene pathways that you want to target. We looked at how some of these small compounds form DNA adducts or cross-link the DNA. So platinum-based ones like cisplatin, carboplatin do that. And so if you have inside of a cell, especially a cell that's in the process of replicating its DNA, so that the 2 strands of the DNA are open, then suddenly you have all these little platinum adducts forming all over the chromosomes, right? And that's just too much for the cell to try to repair. So the cell might undergo apoptosis or just be too damaged so that it cannot successfully turn into 2 daughter cells, right? And that's why these drugs preferentially target proliferating cells, right? Nucleoside analogs, same thing. These types of drugs, they look a lot like DNA bases, so they get incorporated as a cell is proliferating, as it's copying the template strand of DNA and making a new strand. These nucleotides get used as the raw material for making the new strand, but they're not really the A, T, C's, or G's that the new strand of DNA needs to be made from, right? So this ends up messing up the replicated DNA. So another way then that you've disrupted the daughter cells from a proliferating tumor cell to actually be viable. Right? But we did say that the limitations of this are that they don't discriminate. A cancer cell gets hit with these as well as any other cell in your body. So the cells that are most sensitive are the cells that proliferate the most. That's why your hair is lost as a side effect of chemotherapy. That's why people who are undergoing chemo have an awful time with their appetite and their digestion, right? Because those are the rapidly dividing cells that are part of normal tissues. So we said if you want to develop new drugs, right, there are certain limitations that you have to sort of live by. And that is, for the most part, if you start off doing in vitro experiments on cancer cells in the lab, right, even though the ideal situation would be just to take some primary cells, meaning coming from the living tissue, and putting them right into a dish and doing experiments, right? Because if you look at this, remember this images remind you that the tissue that's taken right from the body might look like this. Here's a prostate tumor. Over here is a colon tumor, right? If you go ahead and if you were to try to actually take some of these cells and put them right into a mouse to grow a tumor in the mouse that you then treat with the drug that you're developing, you know, they still look reasonably like what the tumor looked like from the human patient, right? That's the second set of panels here. But the problem is you cannot do that all the time, right? Because as I said, sure, maybe you're lucky, you get enough tissue from this patient's lung that you can maybe generate maybe 6 or 10 mice, and you do your experiment. And then when the experiment's over, okay, great, you need to follow up on the experiment. You need to what? Generate more mice? So you have to go back and get more tissue. You cannot do that, right? So you're limited by the fact that you have to do this intermediate step of introducing primary cells into cell lines or turning them into immortal cell lines, right? And then you have the convenience of always being able to, order this cell line from the NIH, or you have your own stocks of this cell line in the freezer. You go back, you thaw them out, and you inject those into the mice when you're ready to do your mouse experiments, and the tumors grow. But unfortunately, what? Now the tumors look a lot different than the original tumor that came from the person, right? Because remember that only certain aggressive cells will successfully become cell lines, right? And so you are biasing the growth of these cells for only the most aggressive cells, right? And that might not necessarily be what you want your drug development to be based on. Okay, so that's it. So this is just a kind of summary slide that at the end of all of this, right, this whole semester, I never want you to lose sight about, how cancer and the issue of cancer fits in with the evolution of us, right, as this complex living system, and how it is that our cells from conception to birth throughout our childhood, adulthood, and old age are always dealing with cell proliferation. From one fertilized cell that we all started with, right, now as we sit here in front of our computers, right, we are something like 30 plus trillion cells. So it is remarkable to think that over the many years from conception to us sitting here today, right, that the proliferation of these cells is tightly controlled and tightly managed. Because if that were not the case, right, we would not be sitting here. And throughout that, the regulation of discouraging tumors from forming and cells from transforming and cancer from arising is a constant battle that's built into each one of our cells through its numerous tumor suppressor pathways and through the functions of our immune system. And so I think at the beginning of the semester, I asked you, I think the very first day, what are some things that come to mind when we say cancer? Right? And I think all of the things that you guys mentioned in class were by and large negative things. Like you said, metastases and disease and mortality, right? Death, those types of things. And so what I really hope is over the course of the last, 15 weeks or so, as we've begun to understand all of these complex pathways and mechanisms that we have evolved, right, to fight those bad things, in the process of doing so, I hope you've gained a real appreciation for how amazing it is that we have so many mechanisms in place to actually keep things going right. So I think one of the nice side effects of taking a class about cancer, right, is even though it's focused on this idea of disease, what you're really doing is learning, right, all of the things that we've evolved to properly maintain health and regulation of our body's tissues. Okay, so I'll stop there.